me.
Agora acho que já vai.
Nós não podemos esperar mais porque temos depois outra conferência aqui a seguir. Se não der, nós vamos filmar e depois disponibilizamos.
Já está.
Okay, it works. So presentation.
Boa.
Então, boa tarde a todos por terem vindo a esta primeira palestra das Quintas do Mare. É um ciclo de várias palestras que são realizadas nos vários polos do Mare. Aqui estamos hoje no polo da Madeira e temos connosco para fazer uma apresentação a investigadora Sónia Gueroun, que vai nos falar sobre medusas. A palestra será em inglês e terá a duração aproximada de um quarto de hora a 20 minutos e no final temos tempo para uma ou duas perguntas, porque a duração total destas palestras é só de meia hora. Portanto, mais uma vez, obrigada for English. Welcome for coming. Sonia will speak about jellyfish, and she is a researcher from Polo Madeira do Mare. Sonia.
Yeah, thank you. So good afternoon to everyone. So just a few words regarding the context of this work. Since 2018, there was a big project started calling Go Jelly. And the point of the project was to find different use for jellyfish. So cosmetic, fertilizer, food for humans, and food for aquafeeds. And one of my work in this project was to be able to cultivate species with economical potential. So, which means like to able to reproduce the species, to maintain the broodstock, and to have the best condition to rear the species. So the output of all the experiment is for one point to be able to rear the species in the lab, but also all the experiment give also information about the ecology of the species. So for now, regarding the genus of Catostylus tagi, there is 10 recognized species over the world. 3 of them are already exploited, either for aquariums or for exhibition or for fisheries. And less than the 3, we only know the life cycle of only one species. So for ecological point of view, we really lack a lot of knowledge regarding this part. So, question, why we work on Catostylus tagi? First, there is studies show that Catostylus tagi is an edible species. So this can be very economically important, with an economic potential for the countries. And since the species is very known in Portugal, we don't know, for example, the impact of the species could have on the ecosystem. So to do this, we can do field survey, but you also need to do experiment in the lab. So we need to be able to control the life cycle. One of the challenges we had with Catostylus tagi is we started from scratch. There was absolutely no study about the ecology of the species. So basically, our first question was, okay, where the species occurs and when? And for this, we used the citizen science. So instead of doing several years of field research, we just collect data from the citizens. And we know, for example, in the Tigris estuaries, there is a population that occurs all over the year, which means that the medusa stage of Catostylus tagi is quite adapted to several temperature and salinities. So based on this information, we started our work to say, okay, we will work with this kind of temperature and with this kind of salinities. When we work with jellyfish, you need to have an idea about the life cycle. So this is a typical life cycle of a jellyfish. In the top, you have the medusa, which is the sexual stage of the species. They reproduce, giving a larvae that we call planulas. After, this larvae will settle and go to the metamorphosis and give a polyp. This polyp will then reproduce asexually in different ways. And when the conditions are appropriate, they will enter a stage that we call a strobile, and they will release ephira, which is basically a baby medusa. And this one will grow up, give a medusa, and close the cycle again. In our work, we played in 3 parts. We tried to do the fertilization, so to try to find the optimum condition where the fertilization is successful. Then we work on the transition stage from the planula to the polyp, and then from the polyp to the ephira. Thing is, in jellyfish, for the asexual reproduction, there is different way, or I would say mode, in asexual reproduction. Some species will use different mode at the same times. Some species will only use one reproduction way, and the combination of what the species will choose will have a big impact on, for example, on the blooms of your species. So, fertilization first. So for jellyfish, you have several scenarios. Either you have what you call brooding species, which means you have the females and they will keep the larvae inside in the body. In this case, you are lucky. You just have to take the female from the field, you put them in a tank or a container with water, you wait a couple of hours, and after you filter your water and you have planulas. If your species is non-brooding and you know it, you have two ways. Either you take male females in a tank, you wait a couple of days, you cross fingers, and you might have planulas in the end. Or you go for the in vitro fertilization, which means you extract the gonads, you put them in a container with water, and when the conditions are good, you will have planulas after 24, 36 hours. For Catostylus tagi, we had absolutely no idea. And since we don't have time to do like 1 year of survey and to be lucky to find if she's brooding or not, we went for the in vitro fertilization. So we sampled several individuals, we extract the gonads, and under microscope, we distinguish who is a male, who is a female. We select the more mature one. We put the gonads, after cleaning, we put them in the water with the bubbling, and after 24 to 36 hours, we had planulas. So the fertilization was successful. So this was the life cycle of Catostylus tagi. So you have the planula, then you have the polyps, then you have the strobal stage. So this is a stage when the polyps start to transformation to give the ephira. Then you have the ephira and all the development of the gastric system, which is typically for all the jellyfish from Rhizostoma group. So as I said in the beginning, we only know the life cycle of only one species beside tagi, which is Mosaicus in Australia. And as an ecologist on marine biology, it's very important that when you have ephira on your samples, that you are able to identify which species you have. And to distinguish between different species, sometimes easy, sometimes not. For Catostylus tagi, if you want to distinguish between tagi and Mosaicus, it's only few details that you can distinguish between two, which is basically for this genus is regarding the Ropalia shape, the Ropalia canals and the velar canals, which are very different for both species. But beside this, the shape of the ephira is very similar. So we had our ephira, and then we were able to rear them into the young adult stage. So basically, we adapt the size of the tank, depends on the size of the jellyfish, and we give them different kind of meals so they can grow fast. So this is growing in, I would say, standard condition that we use in the aquariums. So we didn't really control, or we didn't know at that stage what are the best conditions. So when we were able to do this part, then we saw the funny part, the experiments. So we knew that we were able to fertilize the species at 18 degrees Celsius and 35 salinities. Now we try to optimize this fertilization. For one point, if someone wants to culture the species so they can know what are the good conditions. And second point, to have an idea, for example, in the wild, when the species reproduce, or at least have an idea. So we played here with different combination of temperature and salinity, but also with light condition. And for several hours, we follow the evolution of the eggs until we got planulas. For now, these are the preliminary results for only 35 salinities. And basically what you can see is when you increase the temperature, you will have more fertilized eggs and you also have faster planulas. So basically we can say, for example, when you have in the feed, so if we have 10 degrees Celsius, maybe it's not the best period when the species will produce. Okay, so we had our planulas. So now we continue to play with the parameters, temperature, and salinity. And here we check the survival of the larvae, how fast the larvae will settle, because it's very important. The larvae, they have very limited amount of energy, I would say. So they need really to settle before they are out of energy, and how fast they can go to the polyp stage. So same thing, we played with temperature salinities, and basically what we saw is that we have very low rate of death. Usually we never exceed 35% of death for the larvae. So for the other conditions, they are very adapted for the condition that you can find in the estuaries. The best condition that we had for the fast settlement and fast polyp development was for 18 degrees Celsius at 35 salinities. But when we increase the temperature, in this case, the best condition was at lower salinities. And for the experiment, we have a significant impact for the temperature and the salinities. Third step, now we had our polyps, and we want to have an idea of how fast the population can reproduce and how fast we can have the first pelagic phase. In this case, we didn't play with temperature and salinity, but we play with temperature and food, which is, from one point, is very important for the culture point of view. So we played with 15 and 20 degrees Celsius, and some treatment received rotifers, some part artemias, and some treatment as control with no food. And for 6 weeks, we checked the survival of the polyps, how the polyps reproduce asexually, so which kind of mode they use, and how much of ephira is produced by strobulation events, and if the polyps give only one ephira, so it's a monostrobile, or multiple strobile, like give several ephiras, knowing that the better the conditions are, more ephira the polyp will produce. So regarding the asexual reproduction, with the conditions that we used, the polyp only reproduce with porocyst, which is ecologically point of view is not the fastest way that the jellyfish or the polyps will increase its population. For example, I would give you an example. If you work with the Aurelia genus, this genus has the ability to combine different kind of reproduction, of asexual reproduction, which is budding, lateral budding, different ways. And just we're starting with few species, with few individuals. For example, we start with 10 individuals. After a couple of months, you will end with 1,000 of them. With Catostylus tagi, is much more complicated. They will only give porocyst, and porocyst is usually considered like a kind of resistant stage. So when you have porocyst, the condition after need to be good, so the porocyst can give you a polyp. So in our culture, we had porocyst, but we didn't see a porocyst giving a polyp, which means if you want to culture the species, you need to do a lot of fertilization so you can have a nice broodstock. And it also gives nice information for the ecology of the species. So for this asexual production, we didn't find any difference, depends of the temperature of the food that we used. There was the same amount of production. And for the second part of the asexual production, the strobulation. Most of the people give several ephira per polyp. And for this case, we found that, for example, the polyps that didn't receive any food, they survived. So for 6 weeks, polyps that didn't feed, they survived very well. They have no mortality, so they are very resistant. And for entering the strobulation process, here when you have higher temperature, the strobulation was faster. As you can see, usually at 18 degrees, after 10 to 20 days, they already start the strobulation process. But at lower temperature, it took them 30 days to start this process. Since the experiments lasted 6 weeks, we couldn't obtain the ephira for the coolest temperature, so 15, because they started the strobulation too late. So we only have results for the 18 degrees Celsius. And for this combination, it didn't matter if we give them ephira, if we give them artemia or rotifer, they produce the same amount of ephira, which is, for culture point of view, is quite good. And the maximum amount of ephira produced by one polyp was 15. So one polyp will give you potentially 15 future jellyfish if they survive after the growth. And as I said, for the different stage, we have either significant impact of temperature or food. So basically, if you want to work with Catostylus tagi, if you want to start your culture in the lab so you can start doing experiments, go for the fertilization, it works. For the preliminary results, you can work with between 18 and 25 degrees Celsius at 35 salinities. Second stage, when you have your larvae and you want to have your polyp, so they can settle fast. If you work at 18 degrees Celsius, go with 35 salinities. But if your temperature is a little bit higher, in this case, decrease your salinity a little bit, so the settlement can be faster and you can have polyps. Then, for the strobulation process, so to have ephira, so baby jellyfish. In this case, with the experiment, but also with the culture that we did beside this, we know that the polyps can give ephira between 15 and 25 degrees Celsius, and they go to the strobulation process continuously between 18 and 25 degrees Celsius, which means if from one point you want to stop to have ephira in your labs, in this case, decrease your temperature, which is also given a very nice idea about how the jellyfish would reproduce in the feed. Because if by decreasing the temperature, we had less ephira, or it takes longer to have ephira, which means that in winter, it's likely that the strobulation process is less, or maybe it doesn't happen in the coolest area. So thank you for your attention. So if you have questions, you are more than welcome. Questions? No. Nice videos.
If there is no questions or comments, we will end this session. And thank you for coming.
Thank you very much.
You can see the other talks of Mare in the site, on the website. So thank you very much.
Thank you. Yes. Stop shared.
Já está a ser transmitido?
No, I can close.
Yes.